Hi, everyone. Happy Friday, and welcome back to this week's Single Cell and Exploration Club. My name is Julie. This is Liam. We're both scientists here about Turing, and due to some last minute changes, I'll be filling in for Hazel for this week. So today I'm excited to show you our analysis today, which we use SmartBulk, which is our platform to analyze bulk RNA-seq data. And on SmartBulk here, we're going to introduce a new feature, WGCNA. So this feature will be readily available very soon for people to try out. So for today's topic, we are going to be decoding the gene expression signatures in chronic hepatitis C and viral resolution. And so Liam here will introduce the biological background and then also take us through the analysis.
Right. Thank you for having me, Julie. So today we will talk about hepatitis C, which is a liver viral infection. Like the title suggests, this hepatitis C virus will inhabit the liver, and over time it will exert damage onto the liver, which will lead to cirrhosis or even liver cancer. Because hepatitis C is symptomless, the patient, when diagnosed, is usually already in a chronic state. And when we talk about the hepatitis C development, this is the trajectory of how the disease progresses. So we start out with a healthy liver that get infected with HCV. It will turn into a stage which we call acute hepatitis. So there's one thing to mention from here, which is that 50% to 25% of the patients will recover themselves from the hepatitis C virus, which is a phenomenon we call spontaneous resolution. Whereas the majority of the patients will get into the chronic state and eventually develop cirrhosis or liver cancer, as I have mentioned. And therefore, today we want to understand how the patients can recover themselves. And we do so by comparing the expression changes between the patients who show spontaneous resolution or clearance versus those who have the chronic hepatitis C. And we would also want to do other comparison as well. So here is our agenda for today. We have different stages of infection, and we would like to do an overview of gene expressions across those stages. We then perform differential expressions across different stages to identify potential changes that are of interest. And lastly, we introduce a new feature that Julie has mentioned, the weighted gene co-expression network analysis, abbreviated to WGCNA on our SmartBulk platform. So for this talk, we introduce a very interesting data set, which can be found in this study right here. The data set consists of total 87 samples, which can be divided into two groups. The chronic are those who never recovered from hepatitis C, and those who showed resolution or clearance. Each of the patients in these two groups were sampled across different time points. The pre-infection, where they did not have the virus, the acute phase, whether it be early or late, and importantly, the chronic follow-up for the chronic patients and the resolution follow-up for the resolution patients. Because we have a lot of stages of hepatitis C here, the first thing we will want to do is to have an overview of expression changes across these conditions, which can be done by the ANOVA feature, which simply perform the ANOVA statistical test. So the ANOVA result on this study will have an output like this, where you can see we have the list of genes on the left, and also their statistical significance on their changes across conditions. So on the right, you can see the position of the selected genes, for example, the PSMB10 here on the chromosome, as well as their statistical information. And down here are the conditions that we have selected for this ANOVA test. We again have the pre-infection, the acute stage, the resolution, or the chronic of the patients. So this ANOVA result is useful when we want to have a quick look at the genes. And so, for example, the PSMB10 here can be seen with more expression during the acute and chronic phase, in which the virus is more active compared to the pre-infection or resolution stages. So perhaps we want to do a more specific comparisons, or we already have a specific comparison in mind, which calls for the differential feature also on our SmartBot platform. So again, in our study, we have different time points, and I believe the most important time point is during the chronic and resolution follow-up, because it will show the most drastic changes between these group of patients, right? So I can take out these two time points in the group of patients and compare them in my differential analysis. To do so, the first thing I do is using the composition feature available on differential expression on SmartBot. So the first level of information I set is the disease status, where you can see I have separated the sample into some spontaneous resolution or clearance and chronic states. And for these two groups that I have separated, I set the next level of information to be the time point where I can select the follow-up or the last stage of the experiment in both of these groups. And therefore, I had group 1 and group 2 for comparison. So the DGE result would look something like this, where you have a list of genes, the direction of expression, and how significant they are in terms of statistical testing. So you can see that the gene CD300H here, when I select it, can be visualized as in this position on the plot, and it has much more expression in the chronic state, in the chronic follow-up compared to the resolution. But perhaps we don't want to assess individual genes. We want to select all the genes that are like CD300H upregulated in chronics and perform a pathway analysis, which is available here on SmartBot differential expression test. By doing so and selected certain databases, we found these pathways that are associated with the upregulated genes in what we found with DE. And also you can see that there are some terms related to hepatitis in our results of enrichment analysis. So we have just compared the chronic and the resolution groups, but perhaps we want to do different kinds of comparison. For example, during the acute phase where we have a lot of viral activity, we also want to investigate them with the pre-infection state. So the first thing I did was to group all pre-infection samples as one. And next, I compare the acute phase in the chronic patients versus that pre-infection, as well as the acute phase of the resolution people, still with the pre-infection stage. Because what we did is two comparisons with the same control group of pre-infection, we may want to find the common genes or genes that are together up or down regulated in both of these comparisons, right? So SmartBot also allows this. So firstly, we again had to dissect our data set so that we have the subset that we want to investigate. So here I have the acute in chronic patients, acute in resolution patients, and the pre-infection block. Then I set the comparison against the control group where I input it as pre-infection. This is extremely useful when you have a lot of groups right here. And by doing so, the SmartBot will automatically assign the control group on this right-hand side comparison panel right here. So the DE test, the two DE tests on acute chronic versus pre-infection, acute resolution versus pre-infection was performed. And we have another feature called Venn analysis that help you to visualize the two comparisons. So on the left-hand side is the circle of resolution acute versus pre-infection, and on the right is the chronic acute versus pre-infection. There's one thing to mention, which is that we have very few significant genes when comparing the acute phase of resolution patients or those who had clearance of virus versus their normal state. So unless this is a mistake on our ends during the pre-processing step, we hypothesize that perhaps their expression profile did not change much during their pre-infection and their acute phase, which can be further investigated. But on the right-hand side, you can see that we see up to 315 significant genes for the chronic, acute versus pre-infections. So there's one more thing with this Venn diagram, which is you can interact with each of the quadrant here to see the genes in them. So for example, I am interested in the four genes that are seen in both of these comparison. I would just click on it. It will be highlighted, and you can see the genes that are shown on the right panel right here. Because there are only a few genes, we can do literature review, and I believe the HLA family genes here have been identified as an antigen presenting gene, which makes sense because it is upregulated in both of these acute phase when compared to pre-infection. Whereas for the quadrants with more genes, which is this one right here, you can click on them and they will show the whole 311 genes, which allow you to do pathway analysis, which calls for a sufficient number of genes. So again, we did pathway analysis and we identified these immune response pathways that are related to the chronic acute comparison with the pre-infection ones. So until now, we have been using the DE results to find the biological pathways, which are a predefined set of genes to explain the changes in phenotypes, right? But here we present a new method, which is called weighted gene co-expression network analysis, abbreviated to WGCNA, which can define gene sets based on our expression profile only and do not use any prior knowledge on existing databases. So just to keep it simple, this is how WGCNA works. The idea is that in our gene expression, there would be genes with similar expression across different samples. And these genes with similar expression across samples are likely to be functionally related. So we can group them into hypothetical group A, group B, and group C, and investigate their biological functions. So what WGCNI did is it has converted our gene expression into a gene dendrogram, where genes are the leaf and genes with similar expression are clustered closer together. And like the gene group that we have on this figure, we can also cluster them using different algorithms on WGCNA. So this cluster of genes with similar expressions and likely to be functionally related, we call them the modules. So using this feature on our initial data set, this is what we have. We have created a dendrogram of genes that by using the default parameters were divided into 46 different modules. However, it should be kept in mind that the number of modules can be adjusted. So I think I will go on to a live demo to how you can see, I can tune the number of modules. So Julie, can you
I do
see
have
like,
yeah, I can see it, but I do have a question at this point. So that other users might also be wondering here. So we see the
yes.
number of modules is 46, and we have a bunch of different colors here. So what do
Hmm.
the gray colors indicate here?
Right, sorry, I missed out on that. So again, I said that we had the modules which on the dendrogram, which are genes with similar expression, right? The colors would be such modules. So you can see all of the genes on the branch of this pink cluster right here belong to the same module. Whereas we would have some parts of the dendrogram with the gray color. These are the background module, or another way to put it is the genes that are not put into any of the module. Those are like the outliers. So thank you for that question. I guess I will go on to the lab demo. Okay, can you see my screen on the lab demo?
Yep, we can see Smart Bulk.
All right. So the way to adjust the number of modules is through the new module cutoff. We have two ways to change the number of clustering. First way is through changing a parameter called deep split. In summary, the higher the deep split is, the more modules you have, and it employs a method called dynamic tree cutting. So perhaps here I see we have up to four, but I believe in the future we might have more or less. I'm not really sure about this, but you can also apply this. And I have already performed it, which you can see here. You would have much more modules. Whereas on the other hand, if you want to have fewer modules, you can either set a lower deep split parameter, or there's another method, which is called static cutting, if I remember correctly. So essentially, it is just cutting the initial dendrogram through their branches. So until you have the desirable number of modules. So perhaps I only want 10 modules or group of genes in my dataset. I will set the threshold to this height and click on apply, which I have already did. And this is the dendrogram that I was left with. So I guess we will stick with this for further analysis. So now we only left with 10 modules. This is an important information. Each of the module is assigned with a value called eigengenes, or another way to call it is eigenmodule. These are the first principal component that summarize the gene expression of a module or a group of genes. So it's like the representative of that module. And SmartBug WGCNA have already clustered the models together to see which models are more similar to each other through this heat map right here. And on the right, you can see different modules and their component genes that make up the module. On this plot here, you can see there is a module eigen-gene correlation. Genes with higher ME correlation are more similar to the eigengene of that module, or the representative value of that module. And therefore, they are likely to be the central genes if this module is in fact associated with a certain traits. So talking about traits, how do we find if a module is associated with a certain trait? We can do that on WGCNA, which is, I believe, is a very promising feature. So all you do is you input a metadata in your dataset that you want to investigate. So for example, I would input the disease status, which have two values. One is chronic and one is resolution, which is my biology question. And I will just click run after naming my test. And this is what I am left with. Again, these are the 10 modules that I am left with after setting a new threshold. And you can see that on the x-axis, there are the two values of the disease status, the spontaneous resolution, and the chronic state. So what WGCNI did is it calculated the correlation of these module with the trait that I have selected. And the higher there are, the more association between that module and the disease state. So here I can see, for example, the module number 0, it has the highest correlation based on color with the state of chronic uh, hepatitis C. So what I can do is I will go back to my module number 0 and see the genes that have the high ME correlation. And I would do more investigation onto these genes. And it should be kept in mind that if you change the number of modules or group of genes with similar expressions, you would have different module trait correlation heat map, right? And you can even pick a better number of correlation than this one. So in summary, let's get back to the presentation. So this is the result of WGCNA, which will be released soon, I believe. So in summary, here I propose two pipelines to understand how the transcriptomic changes between people who have chronic hepatitis C and those who have already recovered from it. One is from using the DE results to find the already defined pathways with already defined gene sets. And the second pipeline is through WGCNA, which defines our own gene sets using our expression data. So thank you for listening. And this is our poll for today. Are you talking, Julie? I can't
Oh, I'm
hear
sorry.
you.
I
Yeah,
was muted this whole time.
right.
So I wanted to take this time to, as the poll is circulating, to explain a little bit more about WGCNA and our motivation behind including this feature on SmartBulk. So WGCNA as a tool can be used as a supplementary analysis in addition to differential expression and also functional enrichment or pathway enrichment analysis. So for differential expression and functional enrichment analysis, these analyses only tell us what genes are expressed or what pathways are enriched in one condition versus another, but it doesn't tell us any information about how these certain genes are connected or correlated to each other. And so this is where WGCNA comes in because WGCNA does give us more info about how these genes are correlated and co-expressed. And from that information, we can draw some conclusions on how these genes are also functionally related. So what I mean by being functionally related, they can be in the same pathway, they can be involved in the same regulatory mechanisms or be in the same complexes. And so WGCNA helps us understand at a deeper level some of the biological insights that we can draw from gene co-expression. And so if you remember, after we group certain genes into modules, we can then perform some further analysis, which can correlate modules with certain phenotypes. So in this way, WGCNA feels kind of similar to pathway enrichment analysis, except the input for WGCNA is only the gene expression matrix, right? The analysis doesn't know anything else about your samples other than the gene expression. So after it finds what genes are co-expressed together and it finds the modules, then you can try to associate what modules are associated with certain phenotypes, certain traits, certain conditions, or treatments, right? And so that's one of the downstream analyses that you can perform with WGCNA. Another thing you can find is that once you have your modules, you can see, well, which modules are correlated with each other. How are these different groups of genes co-expressed with each other or functionally involved with each other? And so I hope that explanation allowed everyone to digest the information a little bit better. And I hope everyone's excited to use this feature on SmartBulk soon. And
I
I guess I'll
believe
take
we
this
have
time.
questions.
Yeah, I'll take this time to address the questions now.
Yeah,
So one person asks, are there any guidelines for choosing how many modules I should choose? So I think this one is kind of dependent on the research question that you want to ask. So let's say, for example, you choose less modules. So when you choose less modules, that means each module will have a larger group of genes, right? So you're kind of looking at like the higher level categorization of these genes, of these modules. And so in that way, each module, since you're having more gene information, maybe it would be a little bit easier to find what phenotypes are associated with a certain module. However, if you go a little bit further down the levels and you have more modules, maybe you'd be able to, I guess, glean more specific information from what phenotypes are associated with those smaller modules. Okay, so I hope that made sense. Okay, next question. Okay, does this platform help in deconvoluting these cell types and look into the expression of those genes? So yes, so this is not a feature that we showed here in the analysis today, but SmartBulk does include a deconvolution feature, in which all you have to do is select the tissue type that your data set is associated with, and it would perform the deconvolution for you. Okay. And then I do have the lead developer of SmartBulk here in the webinar as well, helping us answer some questions. So another person asks, I'm curious to know how you do the metadata harmonization and integration if there are similar multiple public data sets for this analysis. And then so she answered, we implemented the batch effect removal using the Lima method in SmartBulk. And the team is also investigating other tools as well.
That's
Okay.
actually like I plan to have some cell deconvolution run in this talk, but I felt like it was not really relevant, so I left out. But yes, we do support deconvolution. I guess like a very interesting deconvolution test would be to see how the immune cells compositions would be different in the chronic and the resolution state, right?
Yeah, absolutely. Okay. Yeah, and I guess that is also related to the another question a user has, where they ask, can you use results from cell type deconvolution to run WGCNA or cell type specific WGCNA? So if you run the cell type deconvolution, then you'll get a new some new metadata. And so you'll run the deconvolution separately, run the WGCNA separately, and maybe you
Mm.
can see if there are certain modules associated with your certain cell types. We'll give it a couple more seconds for anyone else to um, pop in any questions that they have. Okay, so we have one final question where they ask, how does deconvoluting these cell types compared to single cell RNA-seq in your experiment? So I'm guessing you want to know the differences between the methods in cell type prediction in single cell RNA-seq data and cell deconvolution in SmartBulk. So just in general, uh, what I can say for single cell RNA-seq data, the way we can predict these cell types there is that we use our large public database of single cell RNA-seq data, and we use marker genes from certain clusters to compare against the marker genes within our clusters. And if those marker genes have a high similarity score or a high overlap, then that's how our cell type prediction tool can predict um, the cell types in single cell RNA-seq data. Okay, so we had a clarifying comment, meaning in terms of data consistency. So maybe this one, my colleagues can help answer in the chat as well. If not, we can also follow up with you via email. Okay, if there are no other questions, I think we can end the webinar here. So thank you everyone for joining this week, and we hope to see you all next week. Okay, thank you.